In this video, we'll discuss about metabolism of methionine. It is a essential amino acid and metabolism of amino acid cystine considered together because methionine is ultimately converted into cystine and two molecule of cystine will give cystine. So here this one is essential amino acid. Methionine is essential amino acid. Metabolism of amino acid cystine and cystine considered together because methionine ultimately converted into cystine and two molecules of cystine will give cystine. If you give dietary cystine, that will reduce the requirement of methionine, that is sparing action of cystine and methionine. So the dietary cystine that reduce the requirement of methionine, that is sparing action of cystine and methionine. This methionine utilized for the protein synthesis. It is a glucogenic amino acid. So it utilized for protein synthesis. It is a glucogenic amino acid. It will give glucose. Then glycine, arginine and methionine utilized for synthesis of creatine. Arginine, glycine, arginine and methionine utilized for synthesis of creatine. And methionine is important in various transmethylation reaction and gives different biological product. Methionine is important in various transmethylation reaction and gives different biological product. Now we'll discuss methionine metabolism under three heading. First one is transmethylation and conversion of methionine into homocysteine. So first one is transmethylation, conversion of methionine into homocysteine. Second step in methionine metabolism in cystine synthesis. Third is degradation of cystine. So methionine metabolism, three heading: transmethylation, conversion of methionine into homocysteine, then cystine synthesis, last one is degradation of cystine. We'll discuss step by step. Transmethylation, conversion of methine into homocysteine, methionine into homocysteine. So first one is transmethylation, conversion of methionine into homocysteine, methionine into homocysteine. Now coming to methionine metabolism proper. Methionine metabolism, this is methionine is converted into S-adenosyl methionine, SAM, that is SAM, which is this one. SAM is the universal methyl donor. It is also known as activation of methionine into S-adenosyl methionine. The molecules of ATP recorded here. Here the molecules of ATP is recorded here, and this ATP is converted into inorganic phosphate, converted into inorganic phosphate and inorganic pyrophosphate and inorganic phosphate. So this ATP is converted into inorganic pyrophosphate and inorganic phosphate and gives S-adenosyl methionine. There is transfer of adenosyl group from ATP. Transfer of adenosyl group from ATP to the sulfur of methionine. There is formation of that SAM. So there is transfer of adenosyl group from ATP to the sulfur of methionine. There is formation of SAM, S-adenosyl methionine. This is by the methionine adenosyl transferase. So with the help of enzyme methionine adenosyl transferase. As three energy bonds are utilized here. ATP, three molecules of ATP that make reaction irreversible. Now this S-adenosyl methionine is converted into SAH, S-adenosyl homocysteine, and this reaction is release of methyl group. It release methyl group CH3, which is taken by that acceptor. Take one example. It is taken by the acceptor. Suppose it is norepinephrine is converted into epinephrine. So it taken by the acceptor or it converted into another or important product. This is catalyzed by enzyme methyl transferase enzyme. So it is important. It is known as transmethylation reaction. So this SAM is converted into SAH, S-adenosyl homocysteine. Here in this reaction, release of that methyl group taken by that acceptor. Here acceptor norepinephrine converted into epinephrine. This one catalyzed by enzyme methyl transferase enzyme. This one is the important enzyme. In this reaction, two important enzyme. This one is the first important enzyme. Now this SAH, S-adenosyl homocysteine, with the help of SAH. Now this SAH with the help of homocysteinase enzyme, there is liberation of adenosyl here. So here liberation of adenosyl and it's converted homocysteine. This S-adenosyl homocysteine is converted into homocysteine. So here two reactions are important. First one is methyl transferase. Second one is this methionine synthetase. For this reaction, vitamin B12 is required. In this reaction, there is utilization of methyl tetrahydrofolate. Utilization of methyl tetrahydrofolate, which is converted into tetrahydrofolate. So which this one is converted into tetrahydrofolate. This reaction is utilization of methyl tetrahydrofolate, which is converted into tetrahydrofolate. But vitamin B12 deficiency, suppose this vitamin B12 deficiency, this tetrahydrofolate get trapped as methyl, this one trapped as methyl tetrahydrofolate, and there is functional deficiency of tetrahydrofolate, that is known as folate trap in the deficiency of vitamin B12. So vitamin B12 deficiency, this tetrahydrofolate get trapped as methyl tetrahydrofolate. There is functional deficiency of tetrahydrofolate, that is known as folate trap in the deficiency of vitamin B12. So this is the first step in methionine metabolism, that is conversion of methionine into homocysteine and transmethylation reaction. This one is transmethylation reaction. So step 1, methionine is converted into S-adenosyl methionine. This S-adenosyl methionine, which is universal methyl donor. Then this reaction required ATP. ATP is converted into inorganic pyrophosphate and inorganic phosphate. Transfer of adenosyl group from ATP to the sulfur of methionine. There is formation of ZAM. Then S-adenosyl methionine by methionine adenosyl transferase. 3 energy bond are utilized. Then that SAM is converted into SAH, S-adenosyl homocysteine. Release of methyl group which is taken by that acceptor that norepinephrine is converted into epinephrine catalyzed by methyl transferase. This is called as transmethylation reaction. Then this SAH help for homocysteinase and liberation of adenosyl converted into homocysteine. Homocysteine is converted into methionine with the help of methionine synthetase. That reaction required vitamin B12. And this reaction utilize methyl tetrahydrofolate which is converted into tetrahydrofolate. So vitamin B12 deficiency, there is functional deficiency of tetrahydrofolate, that is known as folate trap. Now coming to step 2, that homocysteine, this one only involved in that second step, the homocysteine converted into cysteine. Second step, homocysteine is converted into cysteine. Now the homocysteine condense with the molecule of serine. So this one condense with the molecule of serine. This gives cystathionine. This one will give cystathionine with the help of cystathionine beta synthetase enzyme. Cystathionine beta synthetase. It is PLP dependent pyriloxyl phosphate dependent enzyme. So this homocysteine condense with serine with the help of enzyme cystathionine beta synthetase and that PLP dependent one, pyroxyl phosphate dependent enzyme converted cystathionine. And with the help of cystathionase, again PLP dependent enzyme, that is cystathionase. This cystathionine is converted into cysteine and homoserine. This homoserine undergo the deamination and decarboxylation. Homoserine is undergoes deamination and decarboxylation. It gives prophanyl coenzyme A. Prophanyl coenzyme A. This prophanyl coenzyme which is converted into succinyl coenzyme A and it is entered into TCA cycle. Via this pathway result in formation of glucose. So this is known as methionine and cysteine. There are glucogenic. So homocysteine condense with serine with the help of enzyme cystathione synthetase form cystathionine. Cystathionase enzyme converted into cysteine and homoserine. This homoserine undergo deamination and decarboxylation. It will form prophanyl coenzyme A. This one will converted into succinyl coenzyme A. This succinyl coenzyme A entered to TCA cycle. Via this pathway result formation of glucose. Now this cysteine molecule synthesis here, it has sulfur. This cysteine molecule have sulfur from that methionine and rest of molecules from that serine. So this is the second step, rest of molecule from serine. This cysteine molecules has sulfur group here, sulfur from that methionine, rest from serine. This one is the second step of methionine metabolism, that is formation of cysteine. Cysteine on condensation with the formation of disulfide linkage which is converted into cysteine. Cysteine is the derivatives of cysteine on condensation of two molecules of cysteine will give cysteine. Two molecules of cysteine will form cysteine. Methionine metabolism step 2 statement form homocysteine. Main one is homocysteine is converted into cysteine. What are the process? Homocysteine condenses with serine to give cystathionine with the help of enzyme cystathionine synthetase. This one is PLP dependent enzyme. Again the cystathione is converted into cysteine. It has sulfur group and homoserine. Then homoserine undergo deamination and decarboxylation to form propionyl coenzyme A. This one will converted into succinyl coenzyme and enter TCA cycle via this pathway formation of glucose. Then that cysteine condenses with formation of disulfide linkage converted into cysteine. 2 molecule of cysteine will give cysteine. Now coming to the third step in that metabolism, that is degradation of cysteine. Here we want to remember that product cysteine. First cysteine is converted into mercaptoethanolamine. Cysteine is converted into mercaptoethanolamine which with pantothenic acid which gives coenzyme A. So this one is pantothenic acid. Pantothenic acid. So cysteine is converted into mercaptoethanolamine which is pantothenic acid which gives this mercaptoethanolamine will gives coenzyme A. So this is the first product. First product given by cysteine. Then second cysteine by the cysteine dioxygenase enzyme. Cysteine dioxygenase enzyme, it give cystic acid and sulfide. Cystic acid and sulfide. This cystic acid is utilized by the production of taurine. Utilized by the production of taurine. It is utilized in the synthesis of bile salt. And so it is important synthetic acid it give by taurine. The sulfide, this sulfide gives in the formation of, this one will gives formation of phosphoadenosine phosphosulfate which is utilized in the monopolysaccharide synthesis and also utilized in detoxification process. So the cysteine, second product acted upon enzyme cysteine dioxygenase enzyme. 2 product cystic acid utilized by production of taurine, utilized synthesis of bile salt. Then sulfide give formation of phosphoadenosine phosphosulfate, utilization of mucopolysaccharide and utilization of detoxification process. Then third reaction, it is converted into pyruvate. Cysteine is converted into pyruvate and this pyruvate will give glucose. So it is glucogenic amino acid. This is the degradation of cysteine. So cysteine will give coenzyme A, then cystic acid and sulfide, then pyruvate, then it will give glucose. These are the product. There are 3 important disorders related to methionine, cysteine and cystine. First important disease, it is cysteinuria. In this disease, there is defect in absorption of cysteine, ornithine, lysine and arginine in the urine. Cola, cysteine, ornithine, lysine and arginine in that urine due to the defective absorption of these amino acid in the renal tubules. This is the first disorder. So due to defective absorption of amino acid in that renal tubules, it is increasing excretion of cysteine, ornithine, lysine and arginine in that urine in the cystinuria condition. Second one is cystinosis. There is accumulation of cystine in the blood and accumulation of cystine in the reticuloendothelial, especially in that lysosome, result in the damage of spleen, liver, bone, etc. Increase of cystine in blood. Third disorder is homocystinuria. It is important. It is due to deficiency of cysteine synthetase. Cysteine synthetase deficiency enzyme. So in this one, due to that increased level of homocysteine in urine and blood, and there is increasing homocysteine level and leads to that cardiovascular disease. First it leads to cardiovascular disease because this homocysteine interfere with collagen formation, because it interfere with collagen formation, collagen cross linking. It interfere with lysyl oxidase, and leads to the aggregation of LDL particles. Leads to that to the coronary artery disease. So these in the diseases leads to increasing homocysteine level. So cardiovascular disease, coronary artery disease, these are the things increasing level of homocysteine. It present as cardiac event also. There is a mental retardation. There is subluxation of lens. So these are the clinical manifestation of homocystinuria. Thanks for watching.